Hi, and welcome to Plant Propagation. If you have any kind of questions that come up during the lectures, during quizzes, any online work, and even during the labs, please give me a text, send me a text, or send me an email. Of course, if we're in-person labs, you can ask me directly. But don't hesitate, especially with the online work, as glitches come up, things aren't always perfectly in line between the lectures and quizzes and things. I do my best, but often there'll be questions that you'll have, and you can always text me, and I respond to you pretty quickly. I'd say like within 24 hours in general, usually a lot faster than that. So feel free. This is an interactive class, and I try to make myself as available as possible to help you learn the material. All right, let's get started. So you can call me Mike and address emails and texts to Mike. That's fine. Let's get started with the material on propagation. So propagating is just increasing the numbers of a species, and you can do that in a couple of different ways. So if you want to propagate animals, you'd breed them. If you want to propagate plants, we can do it in two different ways. Plants can be propagated or cloned via asexual means, so not using sperm and egg like with animals to create a new individual of that species. But asexual is not bisexual reproduction, not with sperm and egg. That is by taking a piece of the tissue of a plant and inducing it through certain conditions to produce new roots and shoots and grow into a whole new plant. Sexual means can also be a way to propagate plants, and that would be by growing plants up until they flower, are pollinated, fertilized, set fruit, and produce seeds. So again, sexual propagation of plants is using seeds. Usually we do that with plants that are annuals, wildflowers, bedding, ornamental flowers, vegetables. Those are mostly propagated through sexual means. Asexual is, again, cloning. We're learning humans are doing it more and more with animals, but mostly it's done with plants. And in the industry of horticulture, asexual cloning is definitely the way that propagation is conducted, because we get exact copies of the parent plants or organisms. Just to start off, I wanted to give you a little history of plant propagation. So we don't know exactly when propagating of plants was started by humans, but humans at some point realized that the wild plants that they were harvesting, and fruits and seeds and other materials they were gathering from plants, that these plants could be manipulated somewhat. So probably some of the first methods were, or one idea of how this came about, that's put forth by an anthropologist named Carl Sauer in the 1950s, thought that humans gathered fruits, wild fruits, and foods, plant foods, and then would bring them back to their dwelling or their village, eat them, and kind of throw out the remains. Let's pretend they were eating a wild pumpkin. And they would eat the flesh and then throw out the seeds and the rest of it kind of in a refuse pile nearby. And at some point, they probably noticed that the same plants that produced those fruits that they gathered in the wild grew then from that refuse pile. So people started to realize they could actually take seeds, and these seeds were somehow the means of the next generation of the plant growing. So seeding was an early plant propagation method. Rhizomes, meaning taking sections of underground stems and moving those about. That was supposed to, both of these things, seeds and rhizomes and root cuttings, were probably some of the earliest propagation of plants that was done, and also led to the whole kind of method of agriculture. At some point, this is what at least the Western world anthropologists say, and there could be many other explanations, but at some point in the distant past, all humans were hunters, hunting animals for food, or gatherers, gathering plants for food, wild plants. And then, like I said, at some point they kind of started to figure out, oh, we don't have to go so far to gather those plants. We can actually gather the seeds and plant them near us, or the roots, or parts of the rhizomes, or bulbs, and move them around. And that kind of gave rise to the idea of agriculture. Jumping way far ahead, or more recently, the mass production of ornamentals started in the 1940s. So why? Why the 1940s? What happened at that point that the market, like people wanted lots of the plants that they saw in magazines or heard about from friends? What was going on at that time culturally? So now we're mostly talking about North America, United States, but probably Europe too. What happened in the United States is around that time, the suburb was created. It actually, these sprawling, very uniform, kind of cookie cutter housing developments first were created, and it was actually like invented. Someone has actually created this idea and implemented it in New Jersey. And these were cookie cutter houses, and they had front yards, and they had ornamental plants. And kind of around that time then is when people started to, the masses of the public wanted to buy plants. And what they wanted to buy were exactly the same things that they saw around in magazines and movies or other people's yards. So that's when cloning became really important in horticulture. People don't go to, for example, to a nursery and say, oh, can I get a rose, please? And the worker says, well, what kind of rose? And people don't say, oh, anything's fine. They say, I want this particular rose, this variety, the one I saw here or over at this park. They want exactly the same thing. So cloning is what is needed, because when you clone plants, the genetics and the phenology, or the form and shape and color, stay the same from generation to generation. Before the 1940s, there was a small horticulture market, but it was for public parks and nurseries and things. It wasn't for the public, so it wasn't a big market. Also around the 40s, air transportation became a commercial thing, and so plants could be transported across the Atlantic and the Pacific. So mostly the Atlantic, but also a lot of early American United States homes and families wanted plants from areas like China, because the whole horticulture industry took off first on the east coast of the United States, and their climate there is close to a lot of climates in China, where they have cold, snowy winters and hot, humid, wet summers. So a lot of our plants, like rhododendrons and azaleas, things like that, they're all Chinese in origin and nativity to certain parts of China. So socially, it became a popular thing to get plants and landscape your yard and make it look pretty. Another interesting thing that happened at the same time was that lawns became a popular thing. And that has a longer history I will explain in a later lecture on turfs. But before lawns and before suburbs, there weren't so many houses on the outskirts of town. There was mostly inner city homes, and those did not have front yards. So the landscaping of the front yard didn't become a thing until after the 40s and 50s. Also, changes and improvements in methods of propagation took place around this time. There was a lot more techniques and knowledge about biochemistry and plant physiology. These cloning techniques like cuttings and things like that became well honed and easily used to produce a lot of plants. And irrigation systems became more automated and more easily purchased and common for normal layperson to get. And then in the last 20, 30 years, tissue culture has become a really new and very important plant propagation method, which we'll talk later about. So let's go on to the outline of this lecture. As I showed you in a couple of slides before this lecture starts out, and this whole course is starting out, by talking about asexual means of plant propagation. propagating using seeds. So sexual reproduction is something that happens when one animal, if you look at animals, produces the sperm, and the other animal, the female, produces the eggs, and those two mate, and the sperm from one and the egg from the other get together and unite to make the embryo. With plants, that's the same as the case for most plants, with a little bit of variation, but sometimes the sperm, which comes from the pollen, and the egg, which is produced in the ovary of a flower, those are both produced in a flower. Sometimes that sperm and egg can come from the same flower. They can self-fertilize. Sometimes that the pollen, which carries the sperm, and the egg and the ovary can be in different flowers on the same plant. That's also self-fertilization, which is common in plants. But also you have cross-pollination, which is more like animals. You have one, for example, like a bush over here that's producing pollen, and that pollen gets to the ovary and egg of on the same species of a bush over there. Not two different individual plants, same species, the pollen and the egg and the sperm and the egg get together. That's sexual reproduction also. What happens when you have sexual reproduction is you have genetics that change because the pollen and the egg are from different sources, and the processes that produce the sperm and the egg, called meiosis, is a cell division process. Those processes involve genetic recombination and mixing. So the progeny, or the offspring, or the children of sexual reproduction are different than the parents. And this is all done with seeds in plants. I'll give you an overview of that, which I kind of just did. Talk about seed anatomy, how to plant seeds, some basic rules of thumb, seed germination techniques and concepts, and some advantages and disadvantages to both. That's what this lecture is about. In future lectures, I'll talk about asexual reproduction, and we'll talk a lot about that because it's cloning is such a big part of the horticulture industry, and it's an easy way for you, even at home, just to propagate your own plants. I switched to a new headset here. So the first few slides, it sounds like I'm talking out of like an airplane hangar or in a tin can. I think these are better. So let's start talking a little more in depth about sexual propagation. As I mentioned, it's the production of seeds. Seeds are the result of sexual reproduction, the fusion of an egg and a sperm. And the offspring or progeny from the parent plants, when they produce the seeds, are unique. They're not exactly genetically the same. So it's not a common landscaping and horticulture industry technique, but for some things like veggies and wildflowers, seeds are, you've purchased probably veggie seeds. A lot of those are, they can be slightly different than the parents, but usually they stay true to their genetics because they've been bred for so long. But mostly horticulture industry uses cloning or asexual reproduction, and that's what we'll talk about the most here. But you can't get away with talking about all this reproductive stuff and cell division and propagation without talking about mitosis and meiosis. And your teacher, me, I'm a plant geek, a science geek, so I love this kind of stuff. And more than just subject you to my geekiness, the way I learn things is sometimes I need to know the details, like all the little parts of the picture to really understand the whole thing. And if you're one of those types of learners, this might help you. If not, bear with me. So there's two types. Think about all organisms, all living things all over the world, all places. They all need a way to create new cells. One cell will divide into two in general. So there's two ways that cell division happens in living things. One is through mitosis, and the other one is meiosis. So mitosis is where, and this is another really silly thing, but mitosis is happening in the somatic cells, meaning somatic means non-reproductive.
So think about, you can think about your own body. Where mitosis, so your toenails are growing, your hair's growing, your skin's replenishing itself, your stomach lining is constantly replacing cells. Where do the new cells come from? They come from the or existing cells. One cell, certain cells in all of those tissues I mentioned, will divide and from one become two, two cells. So that's mitosis, and mitosis is happening in mitoses. Very silly, but it can help you distinguish the two if you forget on a test, like, oh my gosh, what's the difference between mitosis and meiosis? Which one is the reproductive one? Mitosis happens in mitoses. Our toes are not part of our reproductive organs. And that's where, like your toenail cells, one cell divides and makes two new toenail cells that are exactly genetically and morphologically the same. That's mitosis. That's happening all over our bodies except in two places, in the places that produce sperm and the places that produce eggs. So in that process, producing sperm, the male body goes through, the cells go through a process called meiosis. And that's the result of one cell going through meiosis does not end up in two cells exactly the same. It ends up in four cells that are not quite the same. The genetics of each of those four resulting cells are different from each other and from the parent cell, and the number of chromosomes is it's usually, in most organisms, a half of the parent cell. So you've got a cell, there's these proto sperm cells, they're not sperm yet, and that cell divides and divides again. So you get from one parent proto sperm cell, you get four sperm cells, and genetic changes are happening at a number of stages in that meiosis process. So you get four sperm, and they're all genetically unique, and they're all unique and different from the genetics of the parent cells too. So meiosis is in reproductive structures, it is the means of sexual reproduction, and it produces four genetically unique eggs or sperm. And if you see here on the slide, the three places of genetic variation occur in sexual reproduction. Fertilization is one of them. The synapsis and crossing over, the genes and the chromosomes recombine and get mixed up. And these parts called the chromatids, which separate independently. So if you looked really closely at this slide, you'll see meiosis on the left of these little bubbles, and mitosis on the right. So look at the circle in that white graphic on the top left, and it has two chromosomes in it. They look like red and blue things kind of wrapped around each other. That's where all the DNA is. The DNA greatly condensed, makes chromosomes. But you see two chromosomes there. There's four. You've got the red and the blue wrapped around each other on each of those on the left there. And I won't go through all the details, but that divides and makes two new cells, which divide again to make those four sperm cells, and the genetics of each of those form is different, of those four. And mitosis is the cell division process shown on the right of that graphic, and it divides once to make these two cells, but it makes exact copies. That's enough of that. Again, the two types of cell division in plants and all other living things the two types are mitosis, that happens in the non-reproductive tissues, and meiosis, which happens in the reproductive tissues. And in meiosis, the number of chromosomes within the cell is cut in half. So all sperm have only half the chromosome number of their parent cell, and all eggs only have half the chromosome number. So they're getting ready for the sperm and the egg to get together again and form again the full amount of chromosomes to reestablish the full amount. Again, just you don't have to know all the details, but that's how it works. So moving on with describing sexual propagation, in the male parts of the plant are called stamens, and in that stamen is an anther where the pollen is produced, and the pollen contains the sperm. So in sexual propagation, pollination involves the pollen being transferred from the stamen to the female part, the pistil. And the top of the pistil is the stigma. And the stigma receives the pollen, and that's the female part. And that's called pollination. Each pollen grain contains 2 sperm, and fertilization occurs when the sperm are delivered to the egg. Fertilization can, like I said earlier, can occur between the male and the female parts of the same flower, or male and female parts of 2 different flowers on the same plant, or male and female parts of 2 flowers on different plants. The first 2 are called self-fertilization. The last one, involving 2 different plants, is called cross-fertilization. Fertilization occurs then, and seeds are produced. And because each pollen grain in each egg is genetically unique, seed production by a plant produces new genetically unique plants that aren't like the parent. And that can be good if you want to get new variations of things, but it also can be bad if you're trying to get exact copies of the parents and propagate more of the exact same thing. And talking a little bit now about pollination versus fertilization. So you're looking at a little diagram here. You see the flowers kind of cut away. The large white part are indicating the petals. The little yellow round little kernels are actually supposed to represent pollen grains. So pollen is produced there inside the flower, and it has to be released from the flower, and often moved by a pollinator like a bee or some kind of insect will go in there to get the nectar, but it will get pollen on its body or legs often, move to another flower, and inadvertently knock off some of the pollen grains, like in number 4, onto the top of the female part, the pistil. You see 2 little pollen grains that have landed on the pistil. The very top part is called the stigma. So when pollen is transferred from the stamen to the pistil, so produced in number 1, moved through number 2 by some kind of insect pollinator, number 3 onto the pistil, or the very top part of the pistil, that's called pollination, when pollen goes from the male part to the female part. Fertilization is not that same instant. What has to happen, if you look down in number 5, you can look closely, that pollen grain has to, it's like a little seed, it has to germinate, and it sends its little tube down through the pistil, down to that round bulbous part at the bottom there where the eggs are stored. And once it germinates and grows all the way down next to an egg, it will then deliver the sperm that was in the pollen to the egg. That's fertilization. Just like in animals or humans, when the egg and the sperm get together, that's fertilization. The only thing this is a difference in plants in the sense that this process of the time difference between pollination and fertilization can be big. So the pollen can land on the top of the pistil, and a few hours later, that sperm in the pollen tube can grow all the way down and deliver the sperm to the eggs within a couple of hours. But it also can take months. Pine trees in general take 9 months for that pollen grain to germinate, send down its little pollen tube, and deliver the sperm. So the time between pollination and fertilization can be sizable. They're not exactly the same part of the process. First comes pollination, later fertilization. Just different parts of the seed. Once fertilization happens, the seed will start to develop through cell division inside the ovary. By the way, think of something like a sugar snap pea. When you open it up, you've got this long flat green thing. If you opened it up, the 2 sides of it, you'd see the seeds inside. That long green flat thing that we eat and we buy in packages at Trader Joe's, just to snack on, that's an ovary. It's an enlarged, fleshy ovary of that plant. And inside are the seeds that sometimes get fertilized. The seed coat is the outer protective layer of that seed, and it protects the embryo inside from drying out and from injury. The cotyledons, think of a peanut. You take a peanut, you take it out of its shell, which was the ovary, the wall of the ovary, and then you take the peanut out, and it has two halves. Those two halves are cotyledons. They're embryonic leaves. They're not real leaves. They were like leaves inside the embryo, and they contain a lot of nutritive tissue. So if you planted the peanut, or you planted like a kidney bean or some kind of bean in the ground, it opens up, and the first two things you see that sprout and open up are those cotyledons. They provide nutrition to the growing young plant until it can make real leaves that can do photosynthesis. The plumule is just the part of the embryo that turns into the shoot or the stem of the plant. The radicle is the part of the embryo that turns into the root, so it's the embryonic root. And the endosperm supports the growth of the embryo by supplying nutrients and acting as a mechanical barrier. It's around the outside of those cotyledons. So kind of the cotyledon, very loosely, if you think of it this way, the cotyledons are kind of like, well, in a human, the nutrition is coming to the baby through the umbilical cord. But the placenta is a protective layer that's kind of like the endosperm. Yeah, it's a loose analogy, but hopefully it'll give you an idea. By the way, there's a lot of information I'm giving to you, and this is the very first lecture. There's a lot of detail. Sexual reproduction has a lot more kind of science behind it that I'm explaining, and it won't be like this all the time. A lot of the lectures we're going to get into with the asexual means of cloning. We go through some of the basic techniques, and it's very repetitive. A lot of the techniques are the same. It's very simple, not so much, science like naming all the different parts and this like the reproductive process and the parts of the flower. There's a lot of detail I'm downloading, so bear with it. It'll be lighter. And that starting right now, actually, in this part of the lecture, it gets a little bit more simple. So not quite as much to try to remember. And we get into a lot of really practical information in this course, so not so much facts to memorize and terms to remember. In general, when you're planting seeds, the depth of the seed should be the depth of that it should be planted in general should be about twice the diameter of the seed. So if your seed is about 1 inch in diameter, it should probably be planted 2 inches down. If it's like a little poppy seed, just barely under the surface of the soil. The larger the seed, the deeper is planted. So if you have really little seeds like basil, you should just sprinkle them on top of your soil surface and then sprinkle soil on top of them, because you can't really measure putting that twice the diameter of a basil seed. They're just so teeny. So often with really teeny seeds, you bottom water them, like sit the pot or the tray that you've sown those seeds into a tray of water, or at least periodically, so the water soaks up. Because if you like take a watering can or a hose or something, you spray that, these little teeny seeds will just all float away. Direct seeding is a name for the method of planting seeds out into the outside, into the soil of where they're going to grow to maturity. We don't call it indirect seeding, but as opposed to direct seeding, the method that's often used in nurseries and things like that, greenhouses, would be more indirect. It's just germination in pots or flats, places where you're going to start the seeds until they turn into little seedlings, and then you transplant them to the pot or the place that they're going to mature, to the place where you use them or sell them. Often we use these, well, in this course we use these black plastic germination trays filled with some kind of media or soil. The soil in this course and in the industry is called media, And that just, you've heard of like mixed media art, things like that. Media just means some kind of substance that's used for something. And in this case, the reason we don't say soil is often seeds and clones are not propagated in soil. They're done in something alternative that is not just soil. Sometimes it's all perlite, those white little balls you see in potting soil. Or you can propagate seeds and cuttings in 100% sand, or sometimes in pumice, or lots of different things, peat moss. So we call it media. So seeds are germinated in these flat, shallow trays, and then irrigated and kept, often if possible, kept in humid, warm conditions, free of pests, with some good air circulation. That could be in a greenhouse. It could be in this little like hothouse plastic covered tray that you see in the bottom right picture. But some way that keeps the conditions and environment favorable for increasing germination. Germination rate is something that is talked about with seeds and sexual reproduction or propagation of plants. The germination rate is just the percent of the seeds that sprout in a batch. So germination rates are determined by people like you and me that work in these nurseries or research facilities. They'll take 100 seeds of a certain species, a certain batch, and put them in some media and provide the conditions for germination that are known, water, media, humidity, etc., and see how many of those 100 seeds sprouted in a good amount of time. So if only 75 out of 100 sprouted, the germination rate would be 75%. And the germination rate is affected by the viability of the seed. So if only 75 out of 100 seeds sprouted, it could be that some of those were dead. They weren't viable anymore, of those seeds. Or the temperature might not have been quite optimal for all of them to sprout. The moisture level might not have been optimal. Type of plant, some plants don't produce that many viable seeds, and some germinate more easily than others. And the quality of the seed. Sometimes the seeds go bad pretty quickly, or they're old. I know we often get a lot of free seeds from Island Seed and Feed, which is a kind of a farm organic landscaping store north of Santa Barbara, and they just gave us hundreds of seed packets that were from last year, because like veggie seeds and wildflower seed packets, those do not, their germination rate goes down really quickly. So the first year you get them, if they're new, their germination rate is high, about 90% or more. The second year it's about 50%. So no one cares that much when you're just planting a bunch of basil or tomato, but you start to notice when only half of them sprout. So places like the Island Seed and Feed give all their old, 1 year old, they give it all away because the germination rate goes down. This slide is showing you the different stages in germination. Got the seed in the soil. This is showing it half out of the soil. And side note, some seeds, very rarely, most seeds do not require light to germinate. You plant them underground. They need warmth and moisture. But some seeds do require light. You have to germinate them on the surface of the soil. Those are, like I said, rare. It's kind of showing you in this picture. The first thing that pops out of a seed is the radicle. It's the embryonic root, and it starts to form the root, which starts to gather water, which is needed for cell division. And it's utilizing the nutrition that's stored in the cotyledon at this point to produce bigger roots, a stem, and true leaves that are green and can do photosynthesis, which then the plant is able to start making its own sugars and foods and carbs to grow even further. Seedlings is the name of newly germinated plants with only a few leaves and very immature root systems. You don't want to transplant them too soon, but soon after the first true leaves appear, like the one in this picture. And this basil seedling has 4 leaves, and that would be a good time to transplant it. You don't want to wait till the roots get too developed, because if usually in those seeding trays you've got hundreds of little seedlings and they're planted, or they're sprouting really close to each other. If you let them get too old, the roots start intertwining, and then when you try to transplant them, you have to rip them apart, and it's hard on them. So not too young, but pretty soon after they've got 2 to 4 true green leaves present is when you transplant them into a bigger pot. It's called bumping up in propagation terms. Bumping up to a bigger pot. Often you'll put them in a pot with a soil that's more full spectrum. When you're using the media you use to germinate, usually has no nutrition. It's just some kind of thing that holds water, like peat moss or perlite. But once you transplant them, you're putting into a new media that has fertilizer in it too, because now it has a root system. It needs to soak up nutrients from the soil. But be careful. Of course, you don't ever like yank on the leaves or the stems to pull them out to transplant them. You kind of dig them up and sift the roots apart gently so you don't damage them too much. And you'll practice all these things in the lab. Once the plants are transplanted, we then grow them for a while, often in a greenhouse or a hothouse, until they develop a little further, until they're getting to be somewhat mature, usually for a month or so. It depends on the plant, but at least 4 weeks, I would say, at least, even for some just annual veggies and things. But at that point, especially with perennials, you'll want to start the hardening off process. And that means taking a young, somewhat immature plant and moving it out of the conditions that it sprouted in. So you're taking it out of the greenhouse or out of the hothouse and starting to decrease the amount of water that it gets, decrease the amount of humidity in the environment that it's in, and increase the sunlight. So you're taking these young seedlings from this perfect little nursery environment and getting them ready for out there in the big bad world outside, kind of from the inside to the outside, where they're going to receive less water, more sun, and there's less humidity around here at least. So it prepares that young plant for growth and life out external to the greenhouse. One of the reasons often you buy plants, if you do buy them at a nursery, they've been in a greenhouse often their whole life, and then you take them home and they never look quite that good again. So they need this transition period. Their root system gets bigger, gets stronger. They store some more carbohydrates in their stems during the hardening off period, and then when they do get to you plant them outside, they're able to handle it and do well. What are some of the advantages of sexual propagation by seeds? Fast way to get a lot of plants. You can get thousands of seeds and germinate them quickly, easily, for the most part. Not all seeds germinate easily, but a lot of them do. It's easy to do it. There's no special techniques you need to know about, or very few to do to grow plants from seeds. And it's economical. Seeds are quite inexpensive, and you can produce lots of new plants that way. So those are some of the advantages. Disadvantages of sexual propagation. We've already talked about these, but I'll say it again. The new plants that are produced through seeds are not identical to their parents because of the genetics change. And like you're looking here at these, well, I'm not an orchid expert, but they look kind of like cymbidiums. These pink flowers. This is in a greenhouse here that's actually near here in Santa Barbara in Carpinteria, where there's just thousands of the exact same plant clones growing for sale, just like all those orchids you see in Trader Joe's. Like, how can they produce these things and sell them for $4.99? They're cloned, so they're exactly the same, and that cannot be produced. You cannot produce those through seed sexual propagation. So they're not identical to the parents. Sometimes that's good, sometimes that's bad, depending on the industry. Some plants are difficult to propagate from seed. Some require these really elaborate treatments. One of them is fire treatment of the seeds before they'll germinate, and then a cold treatment, and sometimes acid treatment. And it takes longer to propagate things from seed to get them to a mature stage during which you would sell them. So those are some disadvantages. There's pros and cons to it. Lastly, for this lecture, I just wanted to show you a few slides of just really interesting methods of production in the horticulture industry. I call it high-end horticulture. Here's an example of melons that are being produced in Japan, and they have small hats on them to shade them so the skin of that melon is uniform look all the way around the entire sphere. You can't tell, but each of these melons is only one fruit produced on each plant. So all the other flowers are cut off of this one plant, and all of the food that it produces inside the plant is given to this one fruit to make it the best possible. And then they shade it from the sunlight so you don't get a bleached look on the top of the fruit and it looks uniform all the way around. Just produce these like perfect and perfectly uniform fruits that are maximized in their look and their sugar content. And these are worth more for being so uniform and beautiful. There's a much larger gift-giving culture in parts of Japan, and that's what feeds this market of creating these really high-end fruits or food products. Here's another example of a Japanese gift as an unusual and high-end fruit. These watermelons that are in a cube form, and when they're small, they're put into like I've seen them as plastic cage frame that's cubical shaped, and then the watermelon grows into that same form. The light still gets through and everything, but they grow into that form, and they can be done in plastic see-through forms as well. And as the fruit develops, they take on that mold, and then those fruits can be sold for a good sum. These are about 250 bucks to buy one of these watermelons as a gift. And these are sold in a high-end store in downtown Tokyo, not out in the farms and things like that. These are apples, and same thing, produced in Japan, and when they're immature, they have not reddened up, and these decals are put on them, and so as they redden up, which is due to the reaction of the skin of the apple to the sunlight, part of it stays yellowish, and then these are removed and then sold, again fetching a much higher price than a regular apple would, and making it a very valuable and kind gift to give to someone when visiting them. Probably one of the most interesting of these high-end fruits are Buddha-shaped pears. Again, put the pears, these are latched onto the end of the stem where the developing pear is when it's smaller, and clicked on there, and then the pear fills out the mold, and then you can sell these and eat them. Pretty cool. Thanks for getting through lecture 1, and you can go on to take the quiz on this lecture now.